The first outbreak of the Ebola virus was in 1976, and it caused a global race to figure out what it was. If the world was connected 36 years ago as it is today, we might not be here. Ebola is a type of filovirus. It is a cousin in respect to the makeup of the Marburg virus. Ebola is slightly longer than the Marburg virus, measuring in at 920 nanometers in length and 80 nanometers in diameter, and is twice as deadly. Ebola is an envelope virus that is single-stranded negative RNA. It forms into its unique shape. It has been described as a U, a 6, or a Cheerio with a string attached. The protein VP35 plays a critical role in the viral RNA synthesis of Ebola. Ebola has 7 proteins, 3 of which are vaguely understood, while the other 4 are completely unknown. In Ebola replication, the virus attaches to the receptor of the host cell using a glycoprotein called a peplomer, which is a spike on the virus's envelope that will only bind to specific receptors on a host cell. The virus is then engulfed into the host cell. The Ebola envelope and cellular membranes of the host fuse together and the viral nucleocapsid is released. The virus uncoats and the nucleocapsid is transcribed onto a positive stranded mRNA, which is then translated into viral proteins. Glycosylation occurs, which viral proteins attach to glycans, and the glycoprotein precursors are cleaved into glycoprotein 1 and 2. Then glycoprotein 1 and 2 are assembled to form heterodimers. In the meantime, the negative-stranded RNA is synthesized to be used as a template to create a positive single-stranded RNA. The heterodimers multiply to form trimers that will form the surface peplomers of the emerging Ebola cells. New viral copies are made from the negative single-stranded RNA, and the trimers make their way to the cell membrane. The formation of nucleocapsids and the viral envelope occur when the trimers and new genomes gather around the edges of the host cell in preparation for budding. The newly formed Ebola viruses bud from the host cell, gaining part of the host cell membrane. The mature Ebola viruses are then free and ready to attack other cells, keeping the deadly course of action going. Once Ebola is in your body and replicating in your cells, it starts to attack your blood vessels. This destruction causes leaking from the vessels and increased third spacing into tissues. This increase in free-flowing blood can cause external hemorrhaging from natural orifices. Ebola also interferes with naturally occurring clotting cascade and prevents your body's ability to preserve itself. Last but not least, Ebola messes with your body's immune system, preventing any ability to fight off the infection naturally. There are multiple strains of the Ebola virus. Scientists have discovered 3 strains and are concerned that since Ebola is so sneaky, it is easily mutated into different unknown strains. The 3 known strains of the Ebola virus are Ebola Zaire, Ebola Sudan, and Ebola Reston, which are all named after the locations of their individual outbreaks. Ebola Zaire is the most lethal of the strands, with a mortality rate as high as 90%. Ebola Zaire was the first strand of Ebola discovered in 1976. Lucky for the rest of the world, Ebola in itself is a double-edged sword. It is able to infect and kill an extreme number of people, but since the effects come on so quickly, there is a small window of opportunity to spread geographically. After a week of being infected, Ebola patients will not be able to walk, let alone fly on an airplane across the globe. Where Ebola Reston has no effect on humans, but causes devastating depopulation in colonies on non-human primates. The only documented cases of Ebola in the United States were in 1989 in Reston, Virginia. An isolation center for primates used for medical research started developing strange symptoms, and primates started dying by the dozen. The book, The Hot Zone, is a very informative, well-researched novel that goes into great detail as to what happened during that outbreak. But where does Ebola originate from? This is a question scientists have been asking themselves since the discovery of the virus. Ebola is named for a regional river in the area of the first outbreak in Zaire. There are theories that fruit bats could be the natural carrier. Laboratory observations have shown that bats experimentally infected with Ebola do not die and may play a role in maintaining the virus in the tropical rainforest. Ebola could, for all we know, live in pigs, elephants, antelope, dogs, or porcupines. There's no evidence that Ebola is spread through insect bites. Although non-human primates have been a source of infection for humans, they are not thought to be the reservoir. So, how is Ebola spread? It is through the direct contact with blood, secretions, organs, and other bodily fluids of the infected that Ebola makes its way from host to host. Ebola is also sexually transmitted and can cross the placenta in pregnant women to infect and kill the fetus. Burial ceremonies in Africa are also an opportunity to contract the Ebola virus. The ceremonies commonly involve family members kissing the foreheads and hands of the deceased. There has been some evidence that in Ebola resistant strain, the virus is airborne. During the resistant outbreak, there were non-human primates being infected in separate rooms than the original infected, leading scientists to believe Ebola had traveled through the air conduction system. During the initial outbreak in 1976, nosocomial infection was a major cause of transmission, since the hospital staff did not know what the virus was and what it was capable of. The reuse of metal equipment such as needles and syringes was a common practice. Those at risk for contracting the Ebola virus are people who travel to infected areas, specifically Zaire and Sudan. If you do travel to an infected area, be sure to avoid eating bushmeat, which are monkeys and fruit bats. People who conduct animal research are at an increased risk of becoming infected with the Ebola virus. Medical professionals are also at risk of contracting the virus. There have been no known cases of the Ebola virus infecting and killing humans in the United States. This dramatization depicts one possibility of contraction and what follows after being infected with the Ebola virus. We will be following a young man by the name of Greg while he enjoys a nice sunny day's visit to the local zoo. He is excited to see all of the animals, the monkeys in particular. Little does he know what the monkeys have in store for him. The non-human primates are graceful as they swing around their cage. Such showmanship as they eat and drink for the pleasure of the humans visiting the zoo. Could the Ebola virus be stewing in their blood as the monkeys pose for pictures? What would happen if an infected primate escaped and started chasing people down in the streets, hopping from host to host like it's going out of style and depopulating the world as we know it? The incubation period for the effects of the Ebola virus to turn into Ebola hemorrhagic fever is 2 to 21 days. In a mere 3 weeks, you could go from this to this. The early stages of Ebola hemorrhagic fever are common to many other diseases such as malaria and typhus. Headache, petechial rash, sore throat, fever, weakness, and muscle aches do not bring one to immediately think Ebola. But as the virus proceeds to replicate millions to billions of times in your body, the signs and symptoms become more specific. Red eyes are a significant indicator of Ebola hemorrhagic fever, as well as nausea and vomiting blood, and bloody diarrhea. As the disease progresses, the patient will start to have psychological effects. They'll develop a mask-like facial expression and an almost zombie-like demeanor. Soon the patient's blood vessels will not be able to hold back the infected blood, and hemorrhaging commences from every orifice in the body. Bleeding from the eyes, ears, nose, and mouth are very common, as well as rectal, vaginal, and urethral hemorrhaging. All of this eventually leading to seizures, coma, and inevitably death. Testing for the Ebola virus consists of ELISA and PCR testing, and the virus can be isolated in a cell culture. No vaccine has been found or created to fight against the Ebola virus. The only treatment is supportive, replenishing fluids, keeping electrolytes balanced, maintaining blood pressure, and making the patient as comfortable as possible. Several potential vaccines are being tested, but it could be several years before any are available. Is there a chance to survive an Ebola infection? Why there is a 10% survival rate, scientists do not fully understand. Those who do survive an Ebola infection will have a long and painful recovery, usually involving hair loss, sensory changes, hepatitis, and chronic weakness and fatigue. Let's prevent this disease from wreaking havoc on our planet. Make sure you wash your hands as with any sort of disease control procedures. Stay away from infected areas and avoid contact with infected people. Always wear your personal protective equipment, and remember, if it's hot, sticky, and not yours, don't touch it.